now we will start with the spermatogenesis, in which you need to know that this will be referring to the process which produce the sperm or the spermatozoa. Okay, and you should also know by now that this will occur in the testis. Okay, the testis will be the male reproductive organ, and the male gonad that is responsible for this production of the sperm. Okay, and inside this testis, there will be many seminiferous tubules, and we should also take note that the spermatogenesis occurs specifically inside the seminiferous tubule. Okay, so let's look at one particular seminiferous tubule, and if we cut the tubule like this, by this cross section, we will be able to see that there will be many cells inside the tubule. Okay, and at the center of the tubule will be a lumen. On the outermost part of the seminiferous tubule will be cells that we call as the spermatogonia. The spermatogonia, there will be many. Okay, and usually we will start the story with the embryonic stage, in which before the baby is being born, we know that this will start with the embryo that will then develop into the fetus. So during the development of the embryo, there will be these primordial germ cells. The primordial germ cells will be inside the seminiferous tubules in the testis of the embryo. Okay, of course, the primordial germ cell will be diploid, the same like the ploidy of the embryo itself. Okay, and then the primordial germ cell will undergo the process of mitosis in order to produce the spermatogonium. Okay, and this actually happens when the human male reaches puberty. When puberty is reached, the primordial germ cell will convert into spermatogonium. Okay, one spermatogonium, many spermatogonia. All these cells on the outer part, on the outermost part of the seminiferous tubule will be the spermatogonia. All will be diploid cells. Okay, and this spermatogonium, one spermatogonium, which is diploid, will start to undergo the process of mitosis, in which we need to remember that through the process of mitosis, one cell will produce two cells. Okay, so through the process of mitosis, one spermatogonium will divide to become another spermatogonium. Okay, one cell will become the spermatogonium again. Meanwhile, another cell from mitosis will be called as the primary spermatocyte. Okay, you must understand that the primary spermatocyte is still diploid. Okay, why? Because this is only mitosis. Okay, mitosis will not change the ploidy. So if the spermatogonium is diploid, the primary spermatocyte is also diploid. Okay, but then the primary spermatocyte will continue with the first meiosis, meiosis 1, in which one primary spermatocyte will produce two cells known as the secondary spermatocyte, the secondary spermatocytes. And since meiosis 1 has been completed, the ploidy has become haploid. Okay, so from 2N, now the secondary spermatocyte is N, haploid. And the secondary spermatocyte will continue with another round of meiosis, that is meiosis 2, and this will produce the spermatids. Okay, so one secondary spermatocyte will produce two spermatid. So two secondary spermatocyte will be producing four spermatids, and all will be haploid as well. Okay, and you can see here in this particular picture, let's say we start with the spermatogonia. There will be many spermatogonia over here. There will be many spermatogonia over here, This all will be the spermatogonia. Let us focus on one spermatogonium over here. Okay, this one spermatogonium, which is diploid, will undergo the process of mitosis. Okay, so you should take note that there will be no primordial germ cell in this seminiferous tubule. Okay, why? Because all the primordial germ cell has already been converted into the spermatogonia. Okay, so again, one spermatogonium undergo the process of mitosis, and this will be producing one new spermatogonium and another cell known as the primary spermatocyte. Okay, so one cell undergo mitosis. This will be producing two cells. One is still known as the spermatogonium, and another one is known as the primary spermatocyte, which is still in the diploid condition. Okay, but then this primary spermatocyte will undergo the process of meiosis 1, and this will be producing two cells. Okay, two cells which are known as the secondary spermatocyte. And since meiosis 1 has been completed, the two secondary spermatocytes are now considered as haploid, haploid cells. And the secondary spermatocytes will undergo meiosis 2, in which one secondary spermatocyte will be producing these two cells. Another one secondary spermatocyte will be producing another two cells. So all these 1, 2, 3, 4 cells are known as the spermatid. The spermatids. So there will be 4 spermatids produced over here. And these spermatids can differentiate. These 4 spermatids will undergo the process of differentiation, in which they will differentiate to become another form of spermatids. Okay, but the spermatids over here are still hanging on to the seminiferous tubule, to the wall of the seminiferous tubule. But then the spermatid will further differentiate, another round of differentiation to form the spermatozoa or the sperm, the male gamete. This will be the fully formed spermatozoa, in which we can see the shape is like a normal sperm lah, in which we can see the head, we can see the middle piece, and we can see the long tail. Okay, and this sperm or the spermatozoa will be released into the lumen, of the seminiferous tubule, and later they will go to the epididymis over here. The sperm will be stored for a while inside this structure known as the epididymis. Okay, and you should also take note that in the wall of the seminiferous tubule there will be this cell known as the Sertoli cell. The Sertoli cell. So the Sertoli cell will be the cells that are actually located within the seminiferous tubule. What is so important about the Sertoli cell? The Sertoli cell actually facilitate the spermatogenesis to occur properly, to occur nicely, in which the Sertoli cell will provide nutrients for the sperm. The Sertoli cell will provide mechanical support for the spermatids. Okay, and the Sertoli cell can also provide protection for the spermatid. Okay, and when the spermatids are ready to go, and they will be released from the Sertoli cell in the form of spermatozoa. Okay, and we should know that this spermatogenesis will involve the hormones, and so there will be 5 hormones that you need to mention whenever you want to talk about spermatogenesis. Inside the brain, there will be this gland, a gland known as the hypothalamus. This is a very important gland, sometimes known as the master gland. The hypothalamus in the brain will be able to secrete a hormone known as the GnRH, and the GnRH will go to another gland just below the hypothalamus, also in the brain. This gland is known as the anterior pituitary gland. The anterior pituitary gland will secrete 2 hormones, the follicle stimulating hormone and the luteinizing hormone. Okay, what will happen next? These two hormones that are produced in the brain will be released into the blood. Okay, and they will go towards the testes. In the testes, the LH will stimulate another different type of cell. These cells are known as the Leydig cell. When the Leydig cells are stimulated by the LH, the Leydig cell will be secreting, will be releasing the hormone testosterone. And when the testosterone is secreted, this will increase the process of spermatogenesis. This will stimulate the process of spermatogenesis, in which the Spermatogonia inside the seminiferous tubule will change into the primary spermatocyte. Then it will change to become the secondary spermatocyte. Then they will change into the spermatid. Then they will differentiate to become the spermatozoa. So in other words, when the testosterone is released, sperms will be produced. Okay. At the same time, when the Sertoli cells are stimulated by the FSH, the Sertoli cells will support the spermatogenesis. The Sertoli cell will facilitate the process of spermatogenesis. How? By providing nutrients, by providing protection, by providing mechanical support. Okay, so all of this will enhance the production of the sperm. Okay. However, sometimes the sperm production is too high, in which this will be a wasting. When the testosterone detect that the sperms are too much, then the testosterone will do something else. The testosterone will be released from the testes, and it will go into the blood, go towards the brain. In the brain, the testosterone will inhibit the anterior pituitary gland. When the testosterone inhibits the anterior pituitary gland, then the production of FSH and LH will be reduced, and so spermatogenesis will also be reduced. So the level of the sperm will decrease. And the testosterone can also inhibit another thing. Okay, it will go upwards, even upwards. Okay, and it will inhibit also the hypothalamus. Okay, so when the hypothalamus is inhibited by the testosterone, less GnRH will be produced. So when the GnRH is less, okay, the anterior pituitary gland will not be stimulated. In other words, the production of FSH and LH will be reduced as well. Okay, so we can see that the testosterone at one time it can enhance spermatogenesis. However, at one other time, the testosterone can also inhibit the production of the sperm. This happens when there are too much sperm being produced. And we can also see that there is another hormone that can also lower the production of the sperm. This will involve hormone number 5, known as the inhibin. When the Sertoli cell detect there are too many sperms being produced, the Sertoli cells will secrete this inhibin hormone, and the inhibin will be released from the testes, and it will go upwards to the brain, and it will inhibit the anterior pituitary gland. Okay, as we know, this will also reduce the level of FSH and LH. This is actually a mechanism known as the negative feedback mechanism. Okay, the negative feedback mechanism in which it will lower the production of the sperm when the production of the sperm is too much. Okay,